Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump in, if you enjoy what we do, please take a moment to like, subscribe, and share this with your friends and family. It really helps support the Sand Science Experience.
Okay, so today we're embarking on, I think, a really critical exploration. We're looking into a connection that might genuinely surprise you. It's the link between the air we breathe every day, specifically fine particulate matter, and a severe neurodegenerative disorder, Lewy body dementia. We're diving into a groundbreaking research article that meticulously investigates how these invisible air pollutants could be, well, contributing to this devastating condition.
That's right. And our mission today really is to distill the core insights from this study. We want to help you grasp how fine particulate matter, specifically PM2.5, might uniquely trigger Lewy body dementia, or LBD. And crucially, why these findings are so vital for our collective public health. Okay, let's unpack this then. The study kicks off with just an enormous epidemiological analysis. I mean, the scale is huge. With a sample size this massive, what's the single most striking takeaway about PM2.5 exposure and our brain health?
Yeah, the headline here is truly significant. They looked at over 56 million U.S. Medicare beneficiaries, just a massive cohort. And they found a robust, pretty undeniable link. Chronic PM2.5 exposure was significantly associated with first hospitalizations for what are called alpha-synucleinopathies.
Alpha-synucleinopathies, right?
Yeah.
That's the group of diseases where this specific protein, alpha-synuclein, misfolds, like Parkinson's and LBD.
Exactly. Parkinson's disease, dementia with Lewy bodies, Parkinson's disease, dementia, they all fall under that umbrella.
But it's not just a blanket risk increase for all of these conditions equally, is it? I remember reading there was a critical nuance there.
You've hit on the key point, absolutely. The association was markedly stronger for LBD specifically. So that includes Parkinson's disease with dementia, PDD, and dementia with Lewy bodies, DLB. It was stronger for those compared to Parkinson's disease without dementia.
Okay, stronger how? Were there numbers on that?
Yeah, they calculated hazard ratios. For PDD, the hazard ratio was 1.17, so like a 17% higher risk. And for DLB, it was 1.1, a 12% higher risk. This clearly shows a preferential vulnerability for the LBD spectrum. And, that immediately raises a really important question. Why? Why this particular specificity?
Right, so we've established this strong correlation from this huge population study. But correlation isn't causation, as they say. How did they move beyond that? How do they manage to peel back the layers and try to pinpoint the biological culprit behind this devastating link? What was their sort of smoking gun?
This is where the mechanistic work comes in, using mouse models. They systematically demonstrated an essential role for the alpha-synuclein protein itself, or ASIN as it's often abbreviated. In normal, wild-type mice, chronic exposure to PM2.5 induced some serious effects. Brain atrophy, cognitive deficits, and critically widespread ASIN pathology. Not just in the brain, interestingly, but in peripheral organs, too.
Okay, so the pollution caused the protein pathology in the mice. But how did they definitively prove ASIN was the key player, the mediator, and not just, something else happening alongside?
That's a crucial step. They used genetic knockout mice, so mice where the gene for ASIN was removed. And in these mice, lacking alpha-synuclein, removing it genetically conferred strong protection against all those detrimental effects induced by PM2.5, the brain atrophy, the cognitive issues, the pathology.
Wow, okay, so taking away the protein took away the problem, basically.
Exactly. It clearly established ASIN as an important, maybe even central mediator of this environmental neurotoxicity from the air pollution.
So ASIN is confirmed as the key. But you mentioned the specificity towards LBD earlier. Is it just that PM2.5 causes more ASIN pathology, or does it somehow, almost surgically, target or modify the protein in a unique way that's specific to LBD?
That's exactly the question they tackled next, and this is where it gets incredibly precise, I think. They collected PM2.5 from really diverse global sources in the US, China, Europe, to make sure it wasn't just one location's pollution. And they found this PM2.5 actually induced a conformational change in the ASIN protein. It folded differently. It essentially generated a distinct, entirely new ASIN strain, which they named PM-PFF.
PM-PFF, a pollution-induced strain. And what was different about this strain?
Well, this PM-PFF strain showed accelerated aggregation. It clumped together faster. It was more resistant to being broken down by the cell's disposal systems. It also propagated or spread more effectively between cells and showed increased neurotoxicity. Basically, it mirrored the really toxic ASIN strains that are found in human LBD brains.
So it's not just a general environmental assault causing random damage. PM2.5 is acting almost like a sculptor. actively molding alpha-synuclein into this distinct, hyperaggressive form that seems particularly tuned for LBD's cognitive decline. What happened when they introduced this specific PM-PFF strain into living systems like those mice?
Right, they inoculated it into mice that were humanized to express human ausin. And here's the critical finding. This PM-PFF strain preferentially induced cognitive impairments. This contrasts sharply with what happens when you use standard lab-made ausin fibrils. Those tend to cause primarily motor deficits, more like classic Parkinson's.
That is a crucial distinction, cognitive versus motor.
Absolutely. And it lined up with other data, too. Transcriptomic analyses looking at gene expression showed that both chronic PM2.5 exposure and inoculating with this specific PM-PFF strain caused gene expression signatures that mirrored those found in human LBD, so PDD and DLB. But notably, these signatures did not match those typically seen in Parkinson's disease without dementia.
Okay, wow. That really connects the molecular dots, from the air pollution to the specific protein strain right through to the clinical picture of LBD with its cognitive focus. So bringing it all together, this deep dive seems to provide really compelling evidence, doesn't it? From the massive human data down to these intricate molecular mechanisms. It suggests PM2.5 isn't just some general health threat floating around. It appears to specifically contribute to Lewy body dementia by kind of creating this particular harmful hysin strain.
That's the major takeaway, yes. It identifies a novel environmental mechanism specifically implicated in LBD pathogenesis. And importantly, it highlights this PM2.5-induced ASIN strain, this PM-PFF, as a potential future target for therapies. If we could block its formation or its effects, maybe we could intervene.
Are there limitations here, like with mouse models, or what are the next steps needed in human research to really solidify this?
Of course, there are always next steps. While these mouse models are powerful tools, translating these findings definitively to humans requires more work. We need extensive longitudinal studies in people, tracking exposure and disease development over many years, and ultimately you'd need clinical trials targeting this mechanism. But the immediate takeaway, I think, is undeniable. It underscores the critical importance of continuing research into air pollution's role in all neurodegenerative diseases, and it has profound implications for public health strategies. Reducing exposure becomes even more critical.
Absolutely. A truly thought-provoking exploration of these unseen, yet potentially powerful, connections between our environment and ultimately our brain health. Fantastic insights. Thanks for walking us through that. Remember, everyone, keep learning, keep questioning, and keep exploring. And one last reminder, please like this video and subscribe if you haven't already. It genuinely helps us keep bringing you content here at the Sane Science Experience.